My name is Stephen Park. I'm a professor of cancer medicine at Wake Forest School of Medicine. I am a recipient of United to Cure FL grant with partnership with LLS, FLF, and IFLI. So regarding my project, I can go back to sort of my conversation with my patients. So when I discuss treatment options with my patients, I compare cancer treatment to warfare. So traditional chemotherapy is like a bomb; is very effective, but has a lot of collateral damage. In the same way, chemo kills cancer cells very well, but it also damages healthy normal cells at the same time. That's why patients can lose hair and develop severe nausea and vomiting. On the other hand, a targeted therapy is like a smart bomb. It's just as effective, but with less collateral damage. So, over the past couple of decades, we've seen many targeted drugs being developed for various cancers, including lymphoma. Having said that, it turns out that smart bombs aren't always smart. So sometimes targeted therapies can cause serious side effects because they lack precision. Other times they are not effective and cancer cells become resistant because the treatment is not potent enough. So our project aims to overcome these challenges by developing next generation targeted therapy. So first, how can we improve the potency of targeted therapy? That was our question. So if you've seen a spy movie like Mission Impossible, there's always the scene where the main character cuts the wire to deactivate a ticking time bomb at the last second. So tumor cells are kind of like that, and they rely on different wires to activate and survive. So targeted therapy can cut those wires, but it turns out that cancer cells have backup systems. So if you could cut only one wire, they just switch to another. But if you cut both the main wire and the backup wire at the same time, you might be able to eradicate the cancer cell. So we looked into which wires are essential for the survival of follicular lymphoma, especially when they transform into a more aggressive form. So we focus on pathways called EZH2 and MYC, which are commonly activated during the transformation. And we found that by targeting both of these pathways, we could effectively kill follicular lymphoma cells even after aggressive transformation. So our project is currently working on the drugs that target both the EZH2 and MYC pathways. So we talked about improving the potency of targeted therapy, and the second part is improving precision. So to do that, we developed a drug delivery platform using nanoparticles. So what we do is we encapsulate these targeted drugs inside very small particles called nanoparticles. So the drugs are contained and protected. Then we chemically modify the surface of these nanoparticles so that they can navigate through the body and stick specifically to cancer cells without affecting the normal cells. So using this novel technique, we've shown that these drugs can be delivered precisely to the target tumor with the minimal side effects to healthy organs in our mouse models. Yeah, so we've made very promising scientific discoveries in our laboratory and in animal models. But the key is to translate these findings from bench to bedside. That's where this award comes in. It supports our efforts to optimize this next generation targeted therapy platform so that eventually we can bring it to our patients in the clinic. One recent project focused on a pathway called BCL2. So when a BCL2 inhibitor was first developed, we expected that it would be highly active in follicular lymphoma because most of these follicular lymphoma cells have abnormal BCL2. But it turned out that it was not very effective and is not currently being used for treatment of follicular lymphoma. So our group asked, why are these follicular cells, lymphoma cells, resist to BCL2 inhibitors? And we found that these cells use a backup survival mechanism. Then we discovered that we could disable this backup system by targeting a pathway called aurora kinase. So in our mouse model, when we treated follicular lymphoma with the BCL2 inhibitor alone, all mice died within 40 days because of disease progression. But when we combined the BCL2 inhibitor with an aurora kinase inhibitor, so targeting those both pathways, and all the mice with the lymphoma survived more than 100 days with no evidence of disease, which suggests actually a potential cure. So obviously, this is very exciting because we almost never use the word cure in follicular lymphoma. But if we can translate these findings into the clinic, I believe we could offer a new a treatment option that could potentially change the standard of care in follicular lymphoma. And I discussed our project just in follicular lymphoma, but we are actually expanding the same technique and drug delivery platform to other types of lymphomas, such as mantle cell lymphoma.